right, we're going to get our next speaker hooked up. So our next speaker is Ning-Shengji Gao from Oregon State University. And she's going to talk to us today about competition of exoelectrogens in microbial fuel cells.
Okay, everyone. Glad to be here. I'm Ning-Shengji Gao from Oregon State University. And today I'm going to present our lab work, which is about the competition between the exoelectrogens and non-exoelectrogens in microbial fuel cell and how it's related to the current density and Coulombic efficiency in a more engineering perspective. Okay, the primary advantage of MFC is the ability to combine the high energy efficiency and the high substrate consumption rates. So we can see the MFC has a higher energy efficiency than anaerobic digester and also a higher substrate flux than the aerated biofilm reactor. So from this equation, we know that energy efficiency is directly tied to the Coulombic efficiency, which is a representative of the overall percentage of substrate converted to the electricity. And our lab result showed a relationship between the substrate consumption rate and the current density. The current density is representative of the substrate conversion rate to the electricity. So in order to maximize and optimize the Coulombic efficiency and current density, exoelectrogens need to outcompete the non-exoelectrogens in the MFC. So the non-exoelectrogens often include species such as the aerobic bacteria, fermentative bacteria, and methanogens. So in most cases, proton diffusion will limit the kinetics of the microbes. But there are some situations we can break the barrier of the proton limitation by reducing the electrode spacing or increasing the buffer concentration. Therefore, in this case, the current density is primarily limited by the substrate diffusion and utilization following the standard monode kinetics. For the exoelectrogens, as we said before, the substrate consumption rate is representative of the current density, which is around like 15 amps per square meter in optimal condition, in optimal system. And the relationship between the substrate concentration and substrate flux is also the case for the methanogens. As we showed the lab data here, that in single-chamber air cathode MFC, the methane generation is higher when we feed it with 80 mmol acetate than 10 mmol acetate, so that methane generation can be increased at the higher substrate concentration. Since the methanogenesis is not as thermodynamically favorable as the current generation, the max substrate flux will be lower for methanogen than exoelectrogen. Aerobic bacteria can also have the access to the substrate, and their growth is mainly controlled by the oxygen diffusion into the reactor. And previous studies show that increased current density will decrease the dissolved oxygen into the reactor since more oxygen is consumed by the cathode reaction. Therefore, the aerobic bacteria's curve may look like this. Now we have the kinetics of the three types of microbes in the system. Therefore, we can generate a curve showing the Coulombic response based on their kinetics. As we see here, at low substrate concentration, exoelectrogens already reach fairly high current density, but others, like methanogens, is still remaining at lower substrate flux. Therefore, a large portion of substrate is consumed by the exoelectrogen, resulting in relatively high Coulombic efficiency. With adding of substrate concentration, exoelectrogens can reach their max consumption rate, or we can say max current density. But methanogens substrate flux is still increasing, so the Coulombic response will be decreased accordingly. With even further adding of the substrate concentration, all the three types of microbes reach their max consumption rate. Therefore, the Coulombic efficiency tend to be stabilized. When we overlay our data using the CEA design, the Coulombic efficiency fit in this curve pretty well. So we want to see if there are any other factors that can affect the Coulombic efficiency and current density. Then we did a search on Web of Science based on a topic of microbial fuel cell and Coulombic efficiency during the past 10 years, and the top 50 papers were selected as the database. So from the database, we can see a very clear relationship between the substrate type and Coulombic efficiency. So acetate in general can achieve higher Coulombic efficiency than glucose or wastewater. This can also be seen in our lab data, that acetate can achieve high Coulombic efficiency than other substrate. This is fairly obvious that acetate can be utilized directly by the exoelectrogens, and while others need to be fermented prior to be used. For example, glucose. The fermentation procedure of glucose will cause a large portion of electron loss as a form of hydrogen. Though some part of hydrogen can be oxidized and produce current, but still a significant amount of electrons are lost during the fermentation procedure, causing a decreased Coulombic efficiency. Furthermore, if the fermentation procedure is limiting factor, we will have the decreased current density. Another factor we look at is the inoculum type. We can see some sort of relationship between the inoculum type and the Coulombic efficiency across studies, that MFC effluent can achieve relatively higher Coulombic efficiencies than sludge and wastewater. This is probably because MFC effluent has a higher initial abundance of exoelectrons, which can establish the conductive network. And the inhibitory priority effect of the conductive network can resist the colonization of other species, resulting in higher initial current or Coulombic efficiencies. But the influence of the inoculum type may not hold up for long-term operation, since the community structures likely converge similar over time. So reactor designs can greatly affect the overall kinetics of the cell, and many designs are limited by the proton diffusion, or we can see the internal resistance, therefore resulting in decreased Coulombic efficiency and current density. There are many factors we can optimize to decrease the internal resistance, such as the reducing the electrode spacing, increasing the buffer concentration, or eliminating the use of membrane. From this table, we can see by reducing the electrode spacing and increasing the buffer concentration, we can greatly reduce the electrolyte resistance. And also, when compared with two-chamber MFC, single-chamber design can get rid of the huge membrane resistance. This can also be seen in our lab data. When operated at the same condition, CEA with close
session this evening. This afternoon, we continue our session on microbial ecology and microbial electrochemical systems, mixed cultures. I'd like to welcome Krishna Katuri from KAUST. He's a research scientist there, and he's going to tell us about his work with functionalized electrode surfaces. Krishna?
Hello, good afternoon to all. It's too loud.
Oh, that one's on. Is this one on? This one's off.
Sorry.
So your mic. I'm sorry. Okay, go ahead.
Hello, hello.
That's better.
Yeah, yeah.
Oh, let's bring this up for you. We want to be able to hear you.
Yeah. Hello, good afternoon to all. Today I'm going to present my preliminary results on how the surface chemistry of the anode helped us in discovery of new electrogen and its electrochemical behavior. So based on current knowledge, we all know that certain parameters like external resistance, anode potential, substrate, reactor configuration, these all can affect the microbial ecology of the anodic biofilm. And geobacter sulfuridase is the dominant among anodic biofilm. So a single bacterial species capable of driving whole electrogenesis in this system. So the functional redundancies we can't see in these systems like traditional actors like process for driving the COD mole. So our research question is, can we tune MECs or MFCs for enriching novel electrogens in the biofilm? So that is the main our research question. So what we did here is we try to understand how the surface chemistry of anode can affect the electrochemical communication between the bacteria and anode, bacteria and anode interfaces, and at the same time how it can shape the microbial ecology of the anodic biofilm. So that is our research question. So what we did here is we take graphite rod and we incorporate some functional groups on these carbon electrodes through in situ electroreduction techniques. So those functional groups are amino, carboxyl, hydroxyl, alkyl, and unfunctionalized carbon rod used as a control, and we call here as bare electrode. And we did some surface analysis of these electrodes, that is surface charge and contact angle. And based on this analysis, we identified that the functional groups like NH2, COH, OH are hydrophilic, and it has low.
Connect or not? So you have a line of the methane producing rate,
Yes.
methane production. So just the actual data, real data fit to that model or not?
We haven't done that yet. It's just an estimation.
Any more questions? Okay.
Just a small comment on the calculation of your chip's energy. I think that you don't consider the pH, and therefore if you consider the pH, then you have bicarbonate instead of CO2, and this would change a lot in your calculations.
Okay, the chip free energy is based on like pH 7.
Yeah, then you would have bicarbonate, and then you would recommend to recalculate your values.
Okay.
Let's thank our speaker one more time. And I guess we're going to get started a little early. I don't know if they're all set up yet, but maybe we have another 10 minutes, 8 minutes before the poster session starts, so feel free to talk amongst yourselves. What a great session. Thanks, everybody.